Good morning from Germany. We have Dr. Michele Solimena, and we're welcoming everyone to Ask the Expert. It's a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event and we're going to post it on the Sugar Science site, our YouTube channel, shortly after the presentation. If you have questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And again, today we have as our guest, Dr. Michele Solimena, MD, PhD, coming to us from TU Dresden, where he is the professor of molecular diabetology. He's also the director of the Paul Langerhans Institute at the Dresden Helmholtz Center Munich at the University Hospital and Faculty of Medicine of TU Dresden in Germany, the German Center of Diabetes Research. And he has a very long list of credentials. He has been really involved in some seminal work discovering the sort of the mechanics of stiff man syndrome when he was in the lab of Pietro Di Camilli at Yale, and has a very a deep understanding of the beta cell as well as GAD65. So welcome, Michele, and thank you so much for joining us.
Thank you, Monica, for this great opportunity. I really look forward to interact with people. So let me just say from the outset that I'm a cell biologist. I graduated in medicine. As Monica said early on in my life, we have been working on stiff man syndrome, which led us to the identification of the autoantibodies in this rare disease, and then went on to make a connection with type 1 diabetes. As you know now, this is indeed a well known marker for the disease. When I moved out of Pietro's lab, remaining still at Yale, I decided to move from neuroscience indeed to the field of the pancreatic beta cells and diabetes. And that's what I've been doing since then. In 2001, I moved from the States to this city located here in the, I think you may see it. Do you see my screen?
Yes, it's beautiful.
Right. Yes, this is the east part of Germany. So the former, communist side. It's really a beautiful city. It's called the Florence on the Elbe. You can see really is on the banks of this river, and it's famous for its Baroque architecture and also especially music. Here you see the Opera House, where there have been many premiere of work by Richard Strauss and Richard Wagner. So you're most welcome really to visit it. So yeah, this is a brief introduction. Monica, do you want me to move on to the science?
Yes, I think it would be great if we could, you gave us a little thumbnail sketch of your very sort of long and industrious career path. But let's talk about like what the research you're working on right now and this really fascinating paper you've got going.
Yeah, well, first of all, as I said, here I put in this slide the Vitruvian Man of Leonardo da Vinci, because it's really the man in these days at the center of our interest. But before I go there, let me tell you a few things about the beta cell. So the beta cell is truly an amazing manufacturing plant for insulin. It can synthesize up to 3,000 and more molecules of insulin per second. Just to give you an idea, I mean, this is really an astonishing productivity. And we still do not know how this can be managed. But what we have been able to do very recently is, for the first time, to solve in 3D by photon ion beam scanning electron microscopy the entire, let's say, structure of the beta cell. And in this video, actually, we move through the entire beta cell. This is an assembly of 5,000 section each, 4 nanometer thing that we obtained together with a colleague at Genidia Farm, in Virginia. And this actually is the first time that such a resolution of a beta cell entirely has been obtained. So we can really gain quantitative information about the different compartment of the cell. And of course, this can be processed digitally. And here you see actually an entire reconstruction of this beta cell on the right side. On here in orange, the secretory granule, then you peel it off, you see the mitochondria, then you see the Golgi complex, you remove the Golgi complex, you see the 3D network of the microtubule. This is actually the first time that ever the entire network microtubule in a eukaryotic cell, apart from a E cell, has been resolved. And I think that this is actually quite interesting, also, especially for the people working in the field of stem cell, because you may notice normally the microtubule in the textbook irradiate from the centrioles, but what we found is actually only a very minor part, a minority of the microtubule depart from the centriole in an adult beta cell, which means for people interested in achieving the maturation of stem cells into really fully differentiated beta cell, you would have to move from cells that proliferate, of course, they have it connected there. You would need to disconnect it, and vice versa. If you want to, induce replication of the beta cell, you would have to somehow reconnect the microtubular network to the centriole, and we really don't know anything about how this mechanism may be regulated. But of course, but by the way, since the movie you might have seen were jumpy, I really invite you to go to see the original paper where you can see very well the movies. And we are mainly interested in the biology of the secretory granule when it comes to fundamental work. And we set up recently a way to purify them to homogeneity. You can see here the immunopurified secretory granule on beads, and they can be eluted. The paper again is available in the bioarchives, is about to be submitted, but if you already have a preview of that, you're welcome to look at it. But let me come now really to the main study that I would like to tell you about it. And this actually pertains, and what excited about us is pertains deterioration of the beta cell along the progression from normoglycemia to type 2 diabetes. And in order to gain insight into this process, we actually rely on pancreatic tissue removed from a patient who undergo pancreatectomy. And the advantage of this approach is that we have all information about these patients, and prior to surgery, we can also perform a glucose tolerance test, of course, upon informed consent, which means that we can really rely on all data to assess whether these are patients where they are euglycemic, whether people who are have impaired glucose tolerance, or a patient with type 2 diabetes with a certainty. And then what we do, we take the explanted tissue, we immediately fix it in liquid nitrogen, and then we perform laser culture microdissection for omic studies, not only transcriptomic, but also proteomics. And in the meantime, we can also perform histological studies. And our colleague here in our institute, Stefan Speyer, can perform a functional study in terms of secretion with a protocol that he has really pioneered, that today is also using by investigators, for example, in the context of MPOD. Now, what are the key results of this study? I want to give you just a couple of insights of this paper, again, just being published. First of all, what we find is that there is a dysregulation that is progressive, moving from normoglycemia to type 2 diabetes. Here you see that for the first time, we actually can identify genes that are differentially expressed in people with impaired glucose tolerance versus subjects who are non-diabetic. And you can see that here there are some people who have a, let's say, early onset of diabetes, and these are people with a long history of type 2 diabetes. You can see that there's a progressive dysregulation of gene expression. But let me point out another, I think, interesting aspect of this paper. So we analyzed, again, by laser capture microdissection, not only the transcriptomic profile, but also the proteomic profile, again, on laser capture microdissected islets. And you can see that if you look at the transcriptomic profile of this individual, they cannot be segregated based on their gene expression. Basically, there is overlap between the gene expression of islets from non-diabetic subject versus type 2 diabetic subject. The situation is dramatically different when you look at the proteome of these individuals, proteomic analysis that we carried out together with Matthias Mann in Munich's group. You can see that the proteome of individuals who are non-diabetic is very tightly connected, is extremely similar. On the other hand, the transcriptome of the islets from subject with type 2 diabetes is very divergent and actually is much more distant between each other than any of the subjects with non-diabetic are from each other. And this to us come with the hypothesis or the suggestion, strong suggestion, that the route from of the gene expression profiling of the islet from actually talking about protein in this case, non-diabetic to type 2 diabetes is a trajectory that is heterogeneous in different subjects. And therefore, there is no, let's say, differentiation in our view that would take a beta cell back to a precursor state or to another transdifferentiated, by transdifferentiation, another cell type in the islets. They all look very different and conceivably not resembling any cell type physiologically, present at any time. And last things that I want to show you is actually when we look at epigenetic modification that may correlate also with the upregulation of gene expression that I show you, moving from normoglycemia to type 2 diabetes, you can actually see that histone acetylation in the islet of one of these type 2 diabetes subjects here in purple is very much enhanced compared to the signal for acetylated histone in the islet of a non-diabetic subject. Here in green, you see just the signal for insulin. So this is for us truly exciting to provide us insight into what may happen in the case of the diabetic beta cell, a new view, and of course we are very actively continuing this analysis. So lastly, I want to show you my group and also give credit to the people that performed this and other type of study I did not have the chance to present to today, and of course the funding agencies.
This, this is such beautiful work. I'm just blown away by this visualization. And so did you mention that you worked, one of your colleagues was in Virginia at UVA?
No, actually, we collaborated by sending the sample to the colleagues to Shangsu and Arades at the Genilia farm. So we isolated the islets, established the protocol for fixation and staining the sample. And the images were acquired with a FIPSEM microscope by Arades' group that has this unique setup worldwide. They're really absolutely the leader. And then the images that they provided to us, we went on to further analyze it, also using artificial intelligence to extract the information. Where also quantitative information about the relationship of the different organelles, and as I said, about reconstructing the microtubular network. So this is how. So Andreas in my group that has been really pushing this project. He himself has never been at Genilia Farm, but we are in very close contact with them.
It's beautiful work. I think, I guess I would ask, I also am really impressed with the, the data around the T2, the increase in histone acetylation in T2 versus normal. Have you had an opportunity to look at that kind of scenario with type 1?
No, of course, this is something that we would very much like to do. We would actually like to possibility discussing about extending our approach for collection of specimen from pancreatic, from surgical sample from people who undergo pancreatectomy to type 1. Let me say that until now, we have collected sample from over 450 living donors. We routinely, of course, run the assay for autoantibodies against pancreatic islets together with the group of Ezio Bonifacio and Ed Ziegler. Until now, we only identify one among our pancreatitomized patient with type 1 diabetes. But this is something that I think in the future would be very valuable. Every year, there are tens of thousands of people that unfortunately, mostly for pancreatic cancer, undergo pancreatic surgery. There would be an opportunity by screening systematically to identify also people who might have type 1 diabetes or are in the progress maybe to develop it, and collect specimens that can be immediately freezed and analyzed the way we did. I think that this would be a tremendous asset for the community. But of course, it can only be done by merging resources because, as I said, it's like look the needle in the haystack.
Yeah, I'll have to connect you to our friend at Wake Forest, Giuseppe Orlando. He's a pancreatic surgeon that we've had talk to us, and maybe you guys could have a conversation about starting something like that. I think it's really a great idea, of course. I wondered, and I wondered if anyone from the audience, we have a few people here, would like to ask a question either in chat or live. I'm sure there are some questions. I guess I would say with the microtubules involvement here, I mean, it's very obvious that, right, that they are just integral to the whole structure. Is there any way to kind of make or to access some sort of mutation, maybe in a mouse model or other places where there's mutation in the microtubules, and see how that impacts structure?
So far, we haven't done it. Tubulin itself would be very difficult, I mean, not to touch, but there are, of course, many microtubule associated protein that could, in principle, be target. Our reason to study and the microtubule, in particular, also some microtubule associated protein, because we are very interested in the trafficking of the secretory granule, and they use the microtubule, of course, as the highway along which they move. And we have also published that microtubule mediated transport is one of the features that distinguishes the newer made granule from the older one. The newer one move much more often on microtubule, and the others undergo more often secretion relative to the older one. So that's why we are particularly interested in the relationship between the insulin secretory granule and the microtubule. And of course, in the future would be very interesting to see how affecting some of the machinery for this process would impact indeed on insulin secretion.
Yeah, it seems that that's a really fertile ground for inquiry, I would say. It really just, is there, the beta cells have the junctional connection to each other, the alpha cells. Where does that come in? Have you been able to, image two together, or, a group in the cluster in the islet?
Yeah, actually, this is something that we are now intended to explore further. I mean, this analysis that we did now was mainly focused on the single cells, in terms of quantification, again, of the granules in the primary cell in situ, or I mean, not in situ, they were, not single dissociated. I mean, in the we have images of cell in contact with each other that we are now looking, but we do not have, let's say, two full cells in the same image. On the other end, we have some very quite intriguing observation, for example, about the cilia and the cytoskeleton of the cilia, this is something that would be very interesting to look at, and Andreas is actually doing some of this work. I mean, looking at the cell site contact and cilia, but we would need to acquire even more images for that.
Yeah, that'll be really interesting to see. I mean, because we know, right, that, just sort of speaking from my own background, that the beta cell is polarized in some sense, right? It has a apical and lateral side. So it'd be interesting to see how these microtubules orient themselves in that, paradigm. If it changes.
Yeah, well, one interesting thing, I mean, typically also when we think at the microtubular network, we think at a radial, let's say, arrangement, departing from the central, the perinuclear regions. Here in the images that we have, it's clear that most of, or a large part of the microtubule are located near the plasma membrane. They actually run parallel to it. And the point is that conceivably this is to allow secretory granule to move laterally. Once they reach the cell, the cortex, they can move along the plasma membrane, and whenever they encounter a site that is competent for exocytosis, that's when they may undergo secretion. Of course, as you said, in a polarized fashion, as the machinery is mainly located at the basal pole, where vessels, where endothelial cells are located. But let's say there is this arrangement that is beneath the plasma membrane, which is quite peculiar if one compare it to the way typically the microtubule are drawn in the textbook, which is instead the result of the imaging cells that are proliferating, right, where the microtubule are connected with the centriole, and of course they are there because then eventually you have, in the cell division, you have to. So it's we have to think it in another way.
Yeah, it's definitely different than the neuron, right? So that's curious. And then you asked, I mean, I think it'd be so interesting if you guys can dig deeper into the pathology once the beta cells encounter pathology, what happens to that network? It's going to be really interesting.
Yeah, I mean, at the moment, we do not have, evidence that may be also in itself altered. It would not be feasible, probably, I mean, at least to some extent, to do this type of in-depth analysis that we have done here with the isolated islet in this material surgical specimen in terms of imaging. But of course, we are gaining a lot of information in terms of gene expression and pathway, and especially very interesting at the proteomic level, which is which is quite divergent from the image that we have had also in terms of alteration of gene expression from the transcriptomic analysis. The proteomic analysis can really distinguish non-diabetic and type 2, completely segregated. Whereas this, we don't have it at the gene expression. I think that this is going to be really the next frontier. Of course, we have done 5. We will have to do ideally many more and deep into this data.
Yeah, no, it's an altered state. It's not just a little tweak for type 2, as some say, like, oh, you can go back. Looks like there's maybe no going back, right? Once you have all the proteome in place.
Well, I mean, possibly, but on the other hand, I do, we have evidence of reversibility. So the program on one end is scrambled, but on the other end, there must be a way to come back. Sometimes I think about like a big bang where things, move very far apart from each other. But we have to, I think, diet or bariatric surgery tell us there must be a way to bring things conceivably back, and to the point that you saw are the non-diabetic islet at the proteomic level, which are very in sync with one another.
Yeah, well, maybe getting rid of some of that increased acetylation, and then things calm down.
That's right. I mean, I think it's the very disharmonic relaxation of gene expression in different individuals due to although the genetic background, what happened occurs, and at that point, these nuts and bolts, I mean, things that they diverge in different ways, different subject, different other, but eventually I think that maybe there is a way to bring things back in the training of the organ. Somehow the way also stem cells, we don't know how, once they are transplanted and entrained, they acquire maturity by, again, entraining conceivably due to metabolism in the body in a way we cannot yet do to the same extent in vitro. And changing metabolism may do the trick also. We know they do the trick in type 2 diabetes. We just have to learn how. I mean, which is not easy, of course.
No, these are really some fascinating questions. I mean, are you looking for postdocs? I'm thinking that people are going to be listening to this and thinking this is fascinating. I want to go work on this.
We are looking for the moment for PhD. Yes, also we might have some resources for postdocs. So, I don't want to frustrate anyone, but if there is someone who is, excited, please apply. I mean, we'll find a way.
Yeah, and at the same time, the collaborative nature of this project is pretty evident. So if there are people that are interested in collaboration, right, reach out.
Absolutely. We are very happy to collaborate with colleagues, of course, in Europe, but also in the US. As I said, there is a lot of room for, I think, for many people, I mean, to help us, and possibly we can help them. I mean, it's, we have, a gene expression profile, for example, from this subject who are non-diabetic. We have validated the fact that they are truly non-diabetic because we have also an OGTT just a few hours prior to surgery. So we know everything about this subject. And then we do the laser culture microdissection on samples that have been frozen immediately after explantation. So we think that this could be a very valuable resource for a lot of people when they want to know how does the beta cell looks like when it comes to gene expression.
Yeah, absolutely. Well, these are beautiful images and some real functional questions sort of like just unearthed and ready to be, ready to be examined just as a result of these images, I think. And thank you again for joining us. This was really just a pleasure, and I can't wait to share this with the rest of our audience. We do have a lot of people listening via podcast and a lot of watches on our YouTube channel. So I'm assuming that this one will be very popular.
Thank you, Monica. Thank you, Sugar Science. I think it's a great initiative. And yeah, happy someone after wants to drop any question, just email.
That sounds great. Thank you again.
Thank you. Thank you.